Welcome to your daily deep dive into science and health research.
Always so much to explore.
Today, we'll start with a global health challenge.
Hundreds of millions of new cases of bacterial STIs each year. That's according to the World Health Organization.
So many infections go undiagnosed,
Yeah, symptoms can be really subtle.
so it spreads silently,
And that, of course, leads to serious complications.
like infertility.
And miscarriage.
Really highlights the need for better awareness.
Accessible testing, too.
Early detection is key.
Absolutely critical for treatment and to prevent those long-term consequences.
Let's move on to cancer research.
Okay.
There's a new biomarker discovery.
Could be huge, yeah.
It could revolutionize how we predict outcomes.
For certain cancers, right?
Researchers at Fred Hutch,
And MD Anderson are behind this one.
they developed a new technology
And a computational method to go with it to identify this biomarker.
that's showing amazing accuracy.
Predicting outcomes from meningiomas, those are brain tumors.
And breast cancers. Imagine if we could personalize treatment.
Oh, the possibilities!
This could help doctors tailor therapies,
For each patient.
improve their chances of survival.
And quality of life.
It's a step towards more effective cancer treatment.
Absolutely, more targeted.
But cancer cells are tricky.
They are.
They always find a way to thrive.
Even with genetic abnormalities.
There was a study on that
Looking at how cancer cells adapt.
with extra chromosomes.
They adapt and they keep multiplying.
So resilient.
It makes them more resilient, that's right.
We need to understand their strategies
If we want to fight back.
so we can develop better therapies.
Absolutely.
Remember junk RNA?
The genetic material we used to think was useless.
Yeah, turns out it might not be so junky after all.
Not so junky, no. Researchers have found that tRNA introns,
The RNA introns,
they actually regulate protein production.
especially when cells are stressed.
Exact.
So-called junk DNA is actually vital.
For how our cells respond to challenges.
And this is already impacting research,
It is.
especially in breast cancer, right?
Especially in breast cancer, a recent study used FFPE samples,
FFP.
formalin-fixed, paraffin-embedded tissue samples,
Okay.
for RNA sequencing and breast cancer research.
These samples are readily available, aren't they?
They are.
But they haven't been used much for RNA sequencing.
No, not really, but it turns out they're a goldmine.
What did they find?
They can be used to identify immune cell populations and even uncover previously unknown subpopulations of cancer cells.
Wow.
It opens up so many possibilities.
For understanding breast cancer.
And developing more targeted treatments.
From fighting cancer.
To battling infections.
Let's talk about pythiosis.
Pythiosis, rare but serious.
Difficult to diagnose.
And new tests have been developed, in-house enzyme-linked immunosorbent and Western blot assays.
To detect Pythium insidiosum.
The organism responsible for pythiosis.
These are potentially life-saving advancements.
Absolutely.
What makes these assays so special?
They are highly sensitive and specific, really crucial for accurate diagnosis, especially for conditions like pythiosis.
So it allows for timely treatment.
And better outcome for patients.
Now let's look at deep learning in healthcare.
So many studies coming out.
It's transforming the field.
It really is.
There's one in cardiology, using deep learning to analyze genes related to heart failure.
Immunogenic cell death-related genes.
Right, what are the implications of that research?
It could change how we understand how heart failure progresses and maybe even lead to new treatments targeting those specific genes.
Personalized medicine in action.
Exactly.
Deep learning is also being used to assess mental health.
It is.
There's a study looking at major depressive disorder
In older adults.
by analyzing their driving.
Driving behavior is fascinating.
Things like slower reaction times.
Increased lane deviations.
These could be signs of underlying mental health conditions.
Early detection is key, and deep learning can pick up on those subtle cues.
Enabling early intervention for depression.
Yes.
Another study used deep learning to analyze spirograms.
For COPD, chronic obstructive pulmonary disease.
Can they actually predict COPD?
They can. By analyzing the spirometry data, this deep learning model could identify individuals at risk
Before symptoms even appear.
before they exhibit clear clinical symptoms, so it's a proactive approach,
That could slow the disease progression.
and improve outcomes.
Deep learning isn't just for diagnosis, though.
No, it's not.
It can enhance treatment precision.
Especially in radiation therapy.
There's a study using deep learning to estimate radiation doses
to specific organs.
for a certain radiopharmaceutical.
64C dodorituximab.
Okay, and how did that work?
This deep learning model, it accurately predicted the radiation dose to different organs so they could fine-tune the therapy
Minimize damage to healthy tissues.
and maximize the impact on the cancer cells.
Deep learning is even being used in ophthalmology.
For diabetic retinopathy.
They're analyzing retinal images.
To detect the condition.
And how accurate is it?
Highly accurate. Which is great because early detection is key to preventing vision loss.
From cardiology to ophthalmology,
Deep learning is everywhere.
a versatile tool in healthcare.
It's just the beginning.
Now let's talk proteomics.
Okay.
Ultima Genomics is taking on a huge project
Huge. The UK Biobank chose them.
to power the biggest proteomics study ever, using their UG100 sequencing platform
Analyzing over 5,400 protein markers.
across 600,000 samples.
That's incredible.
What are they hoping to learn?
How protein levels change over time, and how that's connected to disease and aging.
This could revolutionize our understanding of human health.
Imagine the possibilities.
Now let's explore RNA sequencing
Sounds good.
and its implications for complex biological processes.
Starting with Ellipsis, a tool for quantifying alternative splicing.
Okay, break that down for me.
Alternative splicing, well, think of a gene like a recipe.
Okay.
Alternative splicing means you can make different dishes from the same recipe.
By choosing different ingredients.
Exactly. So one gene can produce multiple proteins with diverse functions.
And what does ellipsis do?
It analyzes this process
Single cells.
in single cells, and it can pinpoint differences in splicing between healthy and diseased cells.
Powerful stuff.
It's been used to discover novel splicing events in glioblastoma.
That's a type of brain cancer.
Right.
And another study used FFPE samples.
Those formalin-fixed, paraffin-embedded samples.
Yeah, for single-cell RNA sequencing.
Breast cancer research.
They were able to identify immune cell populations.
And a new subpopulation of neoplastic multiciliated cells.
Within breast tumors?
Yes.
That's a significant discovery.
It could lead to more targeted therapies.
Now let's move on to a study on genetic variability,
Okay.
specifically in birth weight.
Interesting.
They used 2 lines of mice,
Bred for high or low variability in birth weight.
and they analyzed their genetic differences.
Using homozygosity by descent mapping.
Can you explain that?
It's like tracing identical twins back to their ancestors, looking for identical gene copies that came from a common ancestor.
So they wanted to see if these identical gene copies influenced birth weight.
Exact.
What did they find?
The results were different between the 2 lines of mice.
So
So it suggests that other factors besides those gene copies are at play.
it's complex.
Genetics is complex.
Let's move on to structural biology.
Okay.
AlphaFold is making waves
It is.
for its ability to predict protein structure.
Developed by DeepMind.
Now they're using it to predict the structures of disordered proteins.
That's right.
Disordered proteins don't have a fixed structure. How could you predict that?
They're like shapeshifters, constantly changing.
But AlphaFold is giving scientists a glimpse into that world.
It's a powerful tool.
What could this mean for research?
Understanding disordered proteins could be key to understanding their roles in diseases and biological processes,
Because they're involved in critical cellular functions.
like signaling and regulation.
This research could lead to new therapeutic targets.
Absolutely.
There's a study that combined computational biology with traditional Chinese medicine.
Interesting combination.
They were looking at tannic acid.
For treating nasopharyngeal cancer.
They used network pharmacology and molecular docking.
To predict how tannic acid interacts with cancer-related targets.
And they tested those predictions in the lab.
They did.
And what did they find?
Tannic acid showed anti-cancer activity.
So it could be a potential therapeutic agent.
It could.
It's amazing how traditional remedies can inspire modern drug discovery.
It is.
Now let's look at a study on premature ovarian insufficiency.
Okay.
They used full-length transcriptome analysis.
To understand the molecular mechanisms.
Can you explain full-length transcriptome analysis?
It's like capturing an entire conversation instead of just snippets.
You get the full picture
Exactly. It captures the complete RNA molecules.
to understand gene expression.
Yes.
So they were trying to understand the genetic conversation happening in these women.
With premature ovarian insufficiency.
And what did they discover?
Specific post-regulatory signatures.
And altered immune cell subsets.
It suggests that dysregulation of gene expression control mechanisms might play a role.
It's a complex condition.
It is.
Now let's explore the unfolded protein response in tuberculosis infection.
Okay.
They used cross-species transcriptomics translation.
Comparing gene expression between humans and the bacteria.
So they were comparing how human cells and bacterial cells respond to stress.
During infection.
And what did they learn?
The unfolded protein response is conserved in both.
In both the host and the pathogen.
That's right.
So maybe we could target this path.
For new tuberculosis treatments.
It's fascinating how seemingly unrelated processes are connected.
It is.
Finally, let's look at a study that used deep learning to analyze whole slide images.
WSOs.
High-resolution images of tissues.
They contain so much information.
And they used a novel approach.
Combining sequence reduction with transformers.
Transformers, those are neural networks, right?
Yes, they're usually used in natural language processing.
So they're applying language models to analyze images.
Exactly.
And how did it work?
It improved the efficiency and accuracy of WSI classifications
Which could impact disease diagnosis.
and treatment.
It's amazing how these different fields are converging.
AI is transforming everything.
It is.
And that wraps up the first part of our deep dive today.
Stay tuned for more groundbreaking research after the break.
Welcome back to our exploration of scientific breakthroughs.
What's next?
Let's dive into biomanufacturing and cell engineering.
Okay.
There's a fascinating study on CHO cells.
HO cells.
They're used to produce therapeutic protein.
Like antibodies.
Monoclonal antibodies, right?
These are essential for so many therapies.
They are, but sometimes they struggle to produce certain complex proteins.
Which hinders drug development.
Exactly.
So what did this study find?
They did a large-scale transcriptomics analysis
Looking at RNA profiles
to understand gene expression. And they identified a new stress biomarker.
in 2HO cells
Yes, this gene is highly expressed when the cells are struggling.
to produce those complex proteins.
Right.
So this could help optimize biomanufacturing
By monitoring this biomarker, scientists can adjust the cell culture conditions
to reduce stress.
and boost protein production.
That could really speed up drug development.
It could.
Speaking of efficiency, there's a study on how cells maintain their structure under stress.
Oh, this is interesting. It's about metabolic cushioning.
Metabolic cushioning.
It's how cells protect themselves from mechanical stress.
How does that work?
They adjust the concentration of certain metabolites.
To act as shock absorbers.
Exactly, to buffer against those stresses.
So it's like their internal protection system.
Right.
And this study looked at how it interacts with cell reproduction.
They used mathematical modeling to simulate those interactions.
Interesting report.
They found that cells can optimize their growth and survival by combining metabolic cushioning with their self-reproduction systems.
It's amazing how cells adapt to challenging conditions.
It is. And speaking of adaptation,
Yeah.
let's talk about plants.
How they defend themselves.
Against pests and diseases.
They don't have immune systems like we do?
No, they don't, but they've evolved other defense mechanisms.
Like what?
This study focused on delimiting strategies.
Delimiting.
It's how we identify and contain the spread of quarantine plant pests and diseases.
Quarantine plant pests. What are those?
These are pests and diseases that could cause serious damage if they spread to new areas.
Like invasive species.
Exactly.
So what did they do?
They used mathematical modeling to assess different delimiting strategies
To see which ones are most effective.
at containing the spread.
That's important for protecting agriculture ecosystem.
It is.
Let's move back to human health.
Okay.
There's a study on a potential new treatment for cancer.
Using PROTACs?
Protacs.
Proteolysis targeting chimeras.
Okay, need a simpler explanation.
They're drugs that use the cell's own machinery to destroy disease-causing proteins.
So they hijack the system.
They do.
And this study developed a computational pipeline.
Called MegaProtac.
What does it do?
It uses molecular docking simulations to predict how effective PROTACs will be.
At forming ternary complexes.
Right.
What are ternary complexes?
It's like a three-way handshake. The PROTAC brings together the target protein and an enzyme that degrades proteins.
So it sets up the target for destruction.
Exactly.
This could accelerate drug development.
By testing designs virtually.
Let's talk about a study on imaging techniques.
This one used fluorescence lifetime imaging mitoscopy.
FLIM.
It measures how long it takes for a fluorescent molecule to emit light.
Okay.
And they developed a method to eliminate autofluorescence.
Autofluorescence.
That's unwanted background fluorescence.
So it's like noise in the image.
Exactly.
And by removing it,
you get a clear picture.
what does this mean for research?
More accurate and sensitive imaging.
Especially for immunofluorescence.
Which is used to visualize specific molecules.
Now let's zoom in on the trabecular meshwork.
In the eye.
The trabecular meshwork.
It regulates fluid drainage in the eye.
Important for eye pressure.
Exactly.
And what did this study find?
They compared gene expression profiles of cultured cells with actual tissue samples.
From the trabecular meshwork.
Yes. And there were significant differences.
In the cellular markers.
Which means that the cultured cells might not fully represent the real tissue.
It's a reminder that cell culture models have limitations.
They do.
Now there's a new data set for studying retinal layers.
Called OCT5K.
OCT.
Optical coherence tomography.
It's an imaging technique.
For the retina.
And this data set contains information on healthy and diseased eyes.
Annotations for multiple diseases, different severity grades.
This will be a valuable resource for researchers.
It will.
There's a study on magnetic particle imaging.
MPI.
A new imaging modality.
They were investigating the impact of image resolution and scanning times on image quality.
Trying to optimize the process. Now let's get back to cancer.
Okay.
There's a study on an interpretable deep learning model of cancer.
Interpretable.
It means we can understand how the model makes its predictions.
Instead of it being a black box.
So we can understand the underlying biology.
That's the goal.
We talked about a study combining computational methods with traditional Chinese medicine.
Tannic acid and nasopharyngeal cancer?
This next study takes a similar approach.
It looks at myrtle extracts.
Myrtle.
Myrtus communis.
Okay.
They studied its antioxidant and anti-tyrosinase properties.
Anti-tyrosinase.
Tyrosinase is involved in producing melanin, the pigment that gives skin its color.
So myrtle extracts could be good for skin health.
Potentially.
And they used a combination of approaches
Phytochemical analysis, bioactivity assays, and molecular docking.
to understand how it works.
Exactly.
Now let's look at a study on a new tool for analyzing genetic data.
De novo peptide sequencing.
De novo.
It means sequencing a peptide without knowing its DNA or RNA sequence.
So figuring out the protein code from scratch.
Right.
And they used a transformer-based framework for this.
Transformers are everywhere.
They are.
And it outperformed existing methods.
For both speed and accuracy.
Impressive.
There's a study on using driving behaviors to identify mental health conditions.
Oh yes, this one looked at major depressive disorder in older adults.
How can driving indicate depression?
Depression can affect cognitive and motor functions, leading to changes in driving habits,
Subtle changes.
like slower reaction times and more lane deviations,
And they use machine learning to analyze the driving data.
and successfully identified individuals with depression.
That could be a valuable screening tool.
It could.
There's another study that analyzed spirogram data.
This time for COPD.
They developed a deep learning model.
That can detect and predict COPD.
From spirometry data,
That's right, it can identify individuals at risk before they even have symptoms.
another early warning system.
Exactly.
Now let's talk about dosimetry,
Symmetry.
measuring radiation doses.
Important for radiation therapy.
They use deep learning to estimate organ-wise dosimetry
For a specific radiopharmaceutical
to make radiation therapy more precise.
and safer.
There's a study on tumor heterogeneity.
The fact that cancer cells within a tumor can be very different. They used mathematical modeling to quantify this heterogeneity.
In tumor spheroids.
3D clusters of cancer cells.
So they're like mini tumors.
In a way,
And what did they learn?
that tumor heterogeneity is significant and affects treatment outcomes.
We need more personalized cancer therapy.
Absolutely.
There's a study on deep learning for diabetic retinopathy
Analyzing retinal images
to detect the condition.
with high accuracy.
Early detection is crucial for preventing vision loss.
It is.
There's another study on deep learning for Parkinson's disease
This one used brain imaging data
to identify individuals with Parkinson's
in the early stage.
before motor symptoms appear.
Exactly.
Now let's look at a study on the mechanical properties of the heart,
Myocardial stiffness.
how stiff the heart muscle is.
Right.
And they used MRI and finite element modeling.
To create a virtual heart.
And what did they find?
Significant variability in myocardial stiffness, both between individuals and within different regions of the heart.
So heart health is very individual.
It is.
There's a study on spatial transcriptomics.
Analyzing gene expression and cell location within tissues,
They developed a new method for understanding how cells communicate.
based on their location and gene expression.
It's like mapping the cellular conversations.
Exactly.
There's a study on a new approach for treating wheezing in children.
Using deep learning to classify wheeze sounds,
They used a transformer-based model
like those used in language processing.
to analyze audio data.
And it worked remarkably well.
Could help doctors make better treatment decisions.
It could.
Now let's talk about multiple sclerosis.
It did.
They used a drug that targets the unfolded protein reformed,
In a mouse model of MS.
and it showed beneficial effects.
It did.
There's another study on a potential new treatment for heart failure,
This one focused on immunogenous cell death.
a type of cell death that triggers an immune response.
They used deep learning to analyze gene expression patterns.
And they identified new targets for therapies.
Right!
There's a study on a potential new treatment for nasopharyngeal cancer,
Using tannic acid
a natural compound.
with antioxidant and anti-inflammatory properties.
And it showed anti-cancer activity.
We did.
Now let's talk about atopic dermatitis.
Eczema.
They targeted a specific protein
IL-31.
with a monoclonal antibody.
And it reduced inflammation and itching?
In a mouse model,
Yes.
there's a study on CAR T-cell therapy for rheumatoid arthritis.
That's usually used for cancer.
They adapted it to target a protein in inflamed joints.
And it reduced joint inflammation in mice.
That's promising.
It is.
There's a study on gene therapy for ALS.
Lou Gehrig's disease.
They delivered a therapeutic gene to motor neurons.
And it slowed disease progression in mice.
That's incredible.
It is.
There's a study on stem cell therapy for spinal cord injury.
They transplanted neural stem cells into injured spinal cords.
Of rats.
Yes.
And what happened?
The stem cells differentiated into new neurons and glial cells,
So there was some regeneration.
potentially paving the way for new treatment.
There's a study on a natural compound for treating cryptococcus
Hinokishal.
from cypress trees,
It showed potent antifungal activity.
both in the lab and in mice.
That's right.
Nature is a powerful source of new therapies.
It is.
And finally, there's a study on diagnosing pythiosis
They developed new diagnostic tests
to detect the organism that causes the infection.
with high sensitivity and specificity.
That's crucial for timely treatment.
It is.
We've covered so much ground today.
We have.
It's amazing how much progress is being made in science and medicine.
It really is an exciting time.
Stay tuned for the final part of our deep dive. Welcome back to the final part of our deep dive into the latest scientific discoveries.
We've explored so much already.
But we still have some ground to cover.
Starting with a natural compound that could potentially treat cryptococcus.
Cryptococcus, remind me what that is again?
It's a serious fungal infection.
Okay.
It can affect different parts of the body, like the lungs and the brain.
Sounds dangerous.
It can be, especially for people with weakened immune systems.
So what did this study look at?
They were investigating hinokitiol as a treatment.
Hinokitiol.
It's a compound found in the wood of certain cypress trees,
Interesting.
and it's known to have antimicrobial properties.
So they're looking to nature for solutions.
Exactly.
Did it work?
Hinokitiol showed potent antifungal activity.
Against the fungus that causes cryptococcus.
Yes, Cryptococcus neoformans.
That's good news.
It could be a valuable new treatment option.
Now let's talk about another challenging infection. Pythiosis.
A rare one, but serious.
Caused by a water mold, right?
Yes, and it can be difficult to diagnose.
So how did this study address that?
They developed new diagnostic tests.
Specifically for pythiosis.
Yes, they created enzyme-linked immunosorbent and Western blot assays.
What's the advantage of these tests?
They're highly sensitive and specific,
So they can accurately detect the infection.
even at low levels,
And that means earlier diagnosis and treatment.
which is crucial for a serious infection like this.
It's amazing how science is constantly finding new ways to fight disease.
It's truly remarkable.
And with that, we come to the end of our deep dive into the latest scientific breakthroughs.
We've covered a lot of ground.
From fighting infections to exploring the potential of new therapies.
It's been an incredible journey.
It has.
Thanks for joining us.
We'll be back tomorrow with another deep dive into the world of science and health.